Hello and welcome back. Thanks for sticking with me. And today we'll be covering monosaccharide metabolism. Okay, so glycolysis. Let's just get through it. It's a way for the body to make ATP. It's one of the most straightforward ways. The first 5 steps involve an investment phase. So it takes 2 ATPs in order to make any ATP in the latter 5 steps. This is the net ATP, that'd be 4. So 1 glucose will produce 2 pyruvate molecules via the catabolic and oxidation reactions, and the final products would be 2 ATP, 2 pyruvates, 2 NADHs. So those 2 pyruvates will go into the Krebs cycle, and I'm not sure if I said it before, but the TCA cycle runs twice per every molecule of glucose, because there are 2 pyruvates produced. So this is classified as an aerobic reaction in the cytosol of the cell, and red blood cells can only do glycolysis and lactate fermentation because there is no mitochondria in these cells. So let's go through the steps. The first step, glucose is phosphorylated by hexokinase, and this takes an ATP. So hexokinase has a broad specificity. It means it can phosphorylate any sort of glucose molecule, any kind of hexo, any hexose would be phosphorylated. So this is in most cells. We have the hexokinases, except for in the liver. It uses glucokinase instead, and it has a different Km. The glucokinase has a higher Km, a higher affinity for glucose, because, of course, the liver stores the glucose, and that means we want more of the glucose being shuttled to the liver for storage. Next, G6P is isomerized by phosphoglucose isomerase into fructose 6-phosphate. And then phosphorylation of F6P by phosphofructokinase into fructose 1,6-bisphosphate also takes a ATP to do, and this is an irreversible step. So the next steps would be past the aldolase step would be ×2, because fructose 1,6-bisphosphate is cleaved into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So with the glyceraldehyde 3-phosphate, you can go directly into the rest of the glycolysis cycle. But DHAP needs to be isomerized first, and then it isomerizes into glyceraldehyde 3P, and then that can go. That's why this latter half of the glycolysis occurs twice. Okay, so glyceraldehyde 3P is oxidized by glyceraldehyde phosphate dehydrogenase, and in this step, it will make an NADH. And an inorganic phosphate would be added, making 1,3-bisphosphate glycerate. And then finally, now we're going to get into the payoff, right? So 1,3-bisphosphate glycerate is going to be dephosphorylated by phosphoglycerate kinase, and in doing so, it will make an ATP. So the phosphate from here is taken and given to ADP. Then it is going to be 3-phosphoglycerate, and that is going to be rearranged into 2-phosphoglycerate by phosphoglycerol mutase. So it mutates. That just means that there's not going to be any change in carbon number. So once there, 2-phosphoglycerate is going to be dehydrated by an enolase. A water is removed, and then it forms phosphoenol pyruvate. Okay, almost done, right? So phosphoenol pyruvate will be dephosphorylated, and then it will be producing pyruvate. And in this location, it shall be dephosphorylated, and that inorganic phosphate would be given to ADP. So there's a lot of steps, and you already have to know the Krebs cycle and the electron transport chain. So how do we make this faster, right? So look where the phosphates are going, right? So steps 1 and 3, phosphates are the phosphates are going to be taken. So ATP is being used up, and here in any of the kinases, the molecules are going to be dephosphorylated in order to make ATP. So finally, we make a pyruvate, and pyruvate has a lot of metabolic fates. It can go through the Krebs cycle and be fully reduced into water. Here you go. It can go into all of this, and then citrate, coenzyme A, then the citric acid cycle. And then it can also go into the fermentation route and making lactate, and that end up as lactic acid. And it can go into gluconeogenesis by being turned into oxaloacetate. And then it can also be transaminated into alanine. So this can even produce one of the amino acids for us. And then it can also make acetaldehyde, and from there go into ethanol. But this is not a reaction of humans, this is a reaction of bacteria. So you don't necessarily have to memorize this chart, but just keep in mind that pyruvate is a flexible molecule. Okay, let's go into oxidative decarboxylation. This occurs in the mitochondrial matrix of the cardiac tissue. Like I said before, the substrate is pyruvate. And from there, pyruvate dehydrogenase will, this is the activation step, remember? So this will activate pyruvate into acetyl coenzyme A, and then from there it can go into the Krebs cycle. This is a very, simplified version, but remember the PDH complex. Oh, and here it is. The pyruvate decarboxylase. So pyruvate will be decarboxylated, and then dihydrolipoyl transacetylase will add the coenzyme A to the newly decarboxylated pyruvate, and then the dihydrolipoyl dehydrogenase will produce the NADH that will go into the electron transport chain. But all in all, this all makes acetyl coenzyme A. It's a protein kinase. Part of it is a phosphoprotein phosphatase, and the source of acetyl coenzyme A for both the TCA and the fatty acid synthesis. So this is an irreversible step. Like I said before, once pyruvate has been converted into acetyl coenzyme A, the cell has no choice but to go through with either like the TCA or the fatty acid synthesis. Okay, so glycolysis is regulated by covalent and allosteric regulation by those irreversible steps. So step 1, that would be the hexokinase. Step 3, that would be the fructose phosphofructokinase 1, and this is also the first committed step. And then finally, step number 10, the pyruvate kinase, right? So hexokinase is inhibited by the amount of G6P because, that means that the cell has a lot of energy. We don't need any more. The first committed step is also known as it is a regulatory step, and it is also known as the it's the rate limited step. Thank you for sticking with me. It was common. The first committed step is the rate limiting step. It's the slowest step. It takes the most energy to overcome the activation and energy barrier, right? So that's why it's the first committed step. And phosphofructokinase 1 is activated by ADP, AMP, phosphofructokinase 2, and fructose 2,6-bisphosphate. So all of these just basically mean that the cell is in an energy deficit. We need to make more. This is also inhibited by high ATP and citrate. High ATP is self-explanatory, and citrate, that means that the TCA cycle is blocked up. So fructose 2,6-bisphosphate also inhibits gluconeogenesis, so that both pathways aren't active at the same time. Makes sense. We don't want to break things down and make things at the same time, because that would be a futile cycle. The hormones, epinephrine, insulin, and glucagon, of course, will affect the glycolysis cycle. Glucagon and insulin will inactivate glycolysis, while epinephrine will activate it. Pyruvate kinase is also inhibited by high levels of ATP, acetyl-coenzyme A, alanine, and CAMP. It is going to be activated by fructose 1,6-bisphosphate in the liver as a feed-forward regulation. So here's a nice chart where this side is glycolysis and this side is gluconeogenesis. So there are about 7 steps that are shared within both pathways, but the irreversible steps, the regulatory steps, are the steps that are not shared between the two paths. So look here, this is what activates glycolysis, or phosphofructokinase, and this is what inactivates it. Here you go. And here. So we want to make glucose when the energy states are low. Okay, so let's go through the math of glycolysis. So step 1, as you remember, it took an ATP in order to phosphorylate something. And in step 2, it also took an ATP. So the first 5 steps are not repeated, remember? So this is only minus 2 ATP. So for step 7, there's going to be 2 ATP made because it's done twice, remember? So same with step 10. It's going to be, of course, repeated as well. And the net yield of one glucose molecule is just 2 ATP. Sounds not very efficient, right? But that's not really the point of just glycolysis. Glycolysis is to make the substrates like pyruvate that goes into something that is a little bit more effective, like a TCA or electron transport chain. So NADH, here you go. This one will make the 3 ATP, but that will go into the electron transport chain, and that would make 5 ATP. So this will be the total ATP production through all of the metabolic pathways, from glycolysis all the way to the TCA cycle. And depending on your textbook and depending on your teacher, this could be like 30 or 32 ATP. It's around that range. You're going to have to pay attention to the way they word the questions and whether they use like the aspartate malate shuttle or the glycerol phosphate shuttle. Remember when we covered that, and that's how the glycerol phosphate shuttle is missing 1 ATP. So depending on that, that's going to like affect the number of ATP yield from a glucose molecule, but in general it's about like 30 to 32. Okay, so fermentation only really occurs in the muscle, and it occurs because of lactate dehydrogenase, and that will oxidize NADH. It reduces pyruvate into lactate in the red blood cells inside the lens, the cornea, the kidneys, testes, and occasionally some white blood cells will do it too. And lactate is a kind of endgame product. Once the body has metabolized glucose. As you can see, it made its its ATP, pyruvate, and then it made its lactate. And lactate itself doesn't actually produce any ATP. It produces the NADH, which can be reduced into NADH, and then glucose will continue again and make another 2 ATP. So this is how our bodies make energy in anaerobic state. And the muscle soreness and the fatigue sets in because there's only a limited amount of glucose. And by the time, like, you go through the cycle so many times, there's only so much glucose and pyruvate available, and lactate will just build up because the muscle cannot process lactate. It needs to be processed in the liver, and our bodies can metabolize lactase back into G6P actually, but it just takes too long. So if you're doing something that's physically active, something that requires a lot of energy, the body's not going to go into its like steady state mode, right? So steady state mode is what I consider like rest and digestion, right? So that only occurs when you're able to relax and go through aerobic fermentation, or not, sorry, aerobic metabolism. But in the meantime, it will build up. So LDH is regulated by substrate concentration, muscle activity, and oxygen concentration. This all works backwards in the liver in the Cori cycle. Okay, so the total ATP made in the red blood cells. So remember, it's going to be -1 ATP for steps 1 and 2. And then for step 7, actually, there's only going to be 1 ATP made. And then for step 10, there's going to be 2 ATP made. And there's no net yield of NADH because, remember in the picture before, it is used up like almost immediately by a lactose dehydrogenase. So you can see pyruvate, lactate, there you go. And this is quite involved. You don't really have to know about this too much right now, but this is why the red blood cells form lactate. Okay, so fructose is a disaccharide, and fructose 1 phosphate will cleave into DHP, and then that goes directly into glycolysis because it can interconvert into glyceraldehyde 3-phosphate. And then in doing so, it bypasses the first regulatory step. It bypasses the hexokinase regulation. So fructose is quite a dangerous molecule because it can just go straight into glycolysis without any regulation, and the rate of metabolism for fructose is much more rapid than glucose. So in doing that, in going through the metabolic path quickly, there is no time for the body to replenish the amount of inorganic phosphate. So it will make a lot of glucose, but it will not be able to make ATP. So the problem is that it will accumulate in the body, and then the inorganic phosphate is not there to make ATP, so only ADP and AMP are left, and those are catabolized because we cannot have these, just lying around. That would cause some toxicity and problems. So it's broken down, and that will cause gout and hyperuricemia. So problematic. And then the accumulation of the fructose will go straight into the fatty acid synthesis, which goes into the esterification process. It goes straight into triglyceride. So this is the process that causes a lot of obesity, especially in the Western world. The way we hydrolyze food and use high fructose corn syrup and store it as a sweetener. There are some healthy alternative sweeteners. I use healthy in quotations because a high amount of fructose, no matter what, is dangerous. Everything needs to be consumed within, moderation. Like agave has a high amount of fructose. Even honey has a high amount of fructose in it. So those I would sweeten things with a little bit of caution. And that is why sugar, like plain old sucrose, is actually the best sweetener because it limits the amount of ATP made. And of course, it's not like I'm not saying only have sugar and you can have as much as you want. Of course not, everything in moderation, but sugar is the best sweetener because the body is able to limit the amount because it goes through the hexokinase regulation. Okay, so mannose is equally dangerous to fructose because it can be isomerized straight into fructose 6-phosphate, and then that goes through the fructose metabolic pathway and once again goes through esterification, makes the triglycerides, and gets stored right in the adipocytes. Most mannose is not really needed in our diets because it is usually like scavenged by hexokinase and recycled. Okay, so gluconeogenesis is the synthesis of glucose from the non-carbohydrate sources such as lactate, glycerol, and amino acids. Pyruvate carboxylase is found only in the liver and the kidneys. Biotin, remember, is its coenzyme. Oxaloacetate is going to decarboxylate and phosphorylate by the PEP carboxykinase enzyme. And those two steps combined will give enough energy to make the PEP, which goes right into glycolysis. Okay, so fructose 1,6-bisphosphate hydrolyzes, once hydrolyzed, bypasses the irreversible phosphofructokinase 1 reaction. So this is just a reiteration of the steps before. In this, phosphofructokinase 1 is a regulatory step, and the dephosphorylation of G6P will bypass another irreversible step, and it will bypass the hexokinase reaction. And we need to bypass this for the activation energy levels of these two regulatory steps are just too high to go like straight backwards. There are, remember when we were going through glycolysis, there were a bunch of reversible steps except for steps 1, 3, and 10. That is why we needed another pathway. This is the pathway to bypass these regulatory steps. So using pyruvate carboxylate, phosphoenolpyruvate carboxykinase, and fructo 1,6-bisphosphatase. Okay, let's go through the regulation. So pyruvate carboxylase is going to be inhibited by acetylcoenzyme A, and it's going to be activated by ADP. Phosphoenolpyruvate carboxykinase is going to be inhibited by ADP, and then these are the steps that are reversible. Fructose 1,6-bisphosphate is going to be inhibited by fructose 2,6-bisphosphate, AMP, and is going to be activated by citrate. And then, it can also be activated by ATP levels. So that means that we have too many, and we want to make glucose. So the G6P in muscles can't be dephosphorylated to make free glucose because we just don't have the enzyme like the one in the liver, and by that I mean like hexokinase. In muscle we have G6P phosphatase, right? And then another regulatory step is that oxaloacetate needs to be reduced into malate before being transported. So it needs to go through a shuttle. Okay, so the PEP carboxykinase synthesis is induced by the glucosteroid hormones and by glucagon. Phosphatase 1 is activated by citrate. It's inhibited by AMP and fructose 2,6 bisphosphate. And G6P is only made in the kidneys and the intestines. So these are the ones that are going to be like exclusive, right? Okay, so the hepatic control is important because basically all extra glucose gets funneled into the liver for storage. G6P is an allosteric activator of PFK1, and it is a competitive inhibitor of the phosphofructose phosphatase 1, as you can see, and that will all kind of play a role in the amount of glucose available for the entire body. Okay, so insulin peaks after we've eaten, and then that is going to inhibit gluconeogenesis. We want to store things, so this is not going to increase the amount of glucose available. Epinephrine works opposite. This is our stress hormone. We need to be active, we need to be alert, we need energies to run away, and it's going to increase gluconeogenesis. Glucagon will inhibit because it works with insulin to store. And in the heart, all these effects are opposite because they have different isozymes. And the heart is just special because it is the one muscle that works continuously. Okay, so this is the end of glycolysis and fermentation. Thank you for your attention and good luck studying.